So my name is Mark McCaslin. I'm the president of Forage Genetics. I'm an alfalfa breeder by training, and what I want to talk about is some of the efforts that are being done in alfalfa breeding and genetics to improve alfalfa as a dairy feed. So I'm going to start by talking about one of the challenges of forages in general. So we've had for a long time plant researchers trying to make forages a better crop for animals, and they kind of have their language doing their thing, and we've got animal scientists then that are working on trying to balance rations, how to feed forages effectively, doing their own thing, and we've got a different language and a different culture, and we haven't done a very good job of talking with one another. And I think the challenge is how do we get plant scientists and animal scientists talking together? So we're designing forages that optimize the potential for forages for improved dairy performance. So I'm going to talk about activities that are being conducted by the Consortium for Alfalfa Improvement. And this started in 2002 when kind of the leaders from these three organizations got together and said, you know what, we're all pretty good at one or two things, but there are gaps. And so the U.S. Dairy Forage Center has been a kind of a center of excellence for ruminant nutrition for a couple of decades. The Noble Foundation is a nonprofit research foundation in Ardmore, Oklahoma, really a world-class organization in terms of altering metabolic pathways and understanding plant metabolism. And Forage Genetics is a vertically integrated alfalfa company doing both breeding, seed production, sales, and marketing. So the three of us got together in 2002 and said, so what could we do to make alfalfa a better crop for dairy animals? So the concept was that we would form an interdisciplinary team, so that means across disciplines from animal science to plant science to breeding, agronomy, and so forth. Would include ruminant nutrition, biochemistry, plant molecular biology, plant breeding, and agronomy. So across those three organizations, we've got expertise in all those fields. And so what we talked about is, so we've got this kind of pooled expertise. So what are logical targets? If we want to make alfalfa a better dairy feed, what are logical targets? And the two, they're fairly obvious. You know, alfalfa is fed primarily as a source of fiber and a source of protein to the dairy cows. So how can we make alfalfa a better source of protein? How can we make alfalfa a better source of fiber? So those are have been the two targets right from the start. So the way we organize the projects is the basis of a lot of this work has been manipulating plant metabolic pathways. So a metabolic pathway is the way a plant essentially takes some precursor and makes an end compound. And by changing various enzymes in the pathway, you can change what the kind of compounds the plant makes. So there's been a lot of basic research done both at the Noble Foundation and Dairy Forage Lab on various pathways that are important in alfalfa quality, forage quality. And then we've been able to target key enzymes to change or key genes to change and create transgenic plants then that have unique characteristics. So once we have those transgenic plants, what we've done then is characterize those for their, we've done biochemical analysis, you know, for example, specific enzyme activity. We've done wet lab compositional analysis. So we made these changes. How does that affect fiber content, for example, fiber composition, protein content, and so forth. Followed by, in most cases, some in vitro analysis. So you take the plant, you put it in rumen fluid, and you see what happens in rumen fluid. That would be an example of the in vitro analysis for fiber digestibility. We take those transgenic plants, we put them in the field. Sometimes you make these changes that look pretty exciting in terms of enhancing forage quality, and they have a negative impact on agronomic characteristics. So an important part of this is you put plants in the field and you see how they perform. And then the final thing was, so once you've got something that looks promising for all of those above things, you say, so what happens when you feed it to a dairy cow? So what I want to walk through is an example of a project that's gone through this whole series of steps. Whoops. So the first one, so when we talked, we brainstormed together and said, so what can we do to improve alfalfa as a source of fiber? The obvious thing is increase the rate of both rate and extent of fiber digestibility. So the culprit in limiting fiber digestibility in alfalfa or in plants, in forages in general, is lignin. So lignin's an anti-quality compound. It's indigestible in its own right, and it binds with cellulose and hemicellulose to make the rest of the fiber less digestible. So we know that in alfalfa, lignin increases with plant maturity. So as the plant gets older, lignin content goes up. And so the goal is, if we reduce lignin, we should be able to both increase the fiber, the digestibility of fiber, and also alter the change in the drop in quality with advancing maturity. So what we want to do is make alfalfa keep its high quality for a longer period of time as it matures. So there's a tool, a genetic engineering tool, that's really perfect for this project, and it's called gene knockout. So what you can do is you can knock out specific genes in a pathway. I'm going to show you a cartoon of what that looks like. So this would be perhaps a typical metabolic pathway where you have compound 1, which gets turned into, in the presence of this enzyme, an enzyme gets turned into compound B, which in turn gets turned into lignin. So that's a very simple example of what a lignin pathway would look like. Each of those genes, each of those enzymes in this example are coded for by a specific gene. And you can use genetic engineering to knock out a specific gene, which means the enzyme isn't formed, and then you end up then reducing the lignin content. So that's a very simple example of what how gene knockout can be used to alter a metabolic pathway. So that's a simple example. This is what the lignin pathway looks like. So there's 12 enzymes, not 2, and it's a branch structure. It's fairly complicated. Our colleagues at the Nobel Foundation now have knocked out every gene for every enzyme in the pathway, and part of what we've done at Forage Genetics then is to characterize the transgenic plants that have resulted from those various knockouts. So what we find is you knock out various genes in the pathway, you have an impact on fiber content, and you also have an impact on fiber composition. So lignin is made up of these 3 kind of subunits, S-lignin, G-lignin, and H-lignin. In alfalfa and corn silage, actually the only ones that are produced in any quantity are S and G-lignin. But by knocking out various enzymes in the pathway, you can change both content and then the percentage of S, G, and you knock out some genes and you get H-lignin form, which isn't normally found in alfalfa or corn silage. So what we've done is compared then these various gene knockouts. And in this slide, this was some of the early work that was done at Nobel Foundation, where they compared, these are various genes that were knocked out. You can see the color bar this is fiber digestibility in situ. Here's the control. So that's normal alfalfa. And you can see the various enzymes have a variable effect on fiber digestibility. So we've compared now all 12 of these knockouts. How do they affect fiber composition, fiber content, fiber composition, fiber digestibility, and agronomic traits. So this is an example. So there's, of all those enzymes we've knocked, of the 12, there's 3 that look promising. And when I say look promising, means they have a significant impact on fiber, on lignin content. They alter lignin composition, and at the same time, they increase fiber digestibility, and they have normal agronomic phenotype. So this is an example of one of these. So I should say, so the green bar here is the transgenic. So that's the reduced lignin line that has a knockout for one of the enzymes, and then the purple bar here is the null. So the null is the control. So that's exactly the same genotype, but without the transgene. So the only difference between these 2 is the presence of this reduced lignin transgene. So no difference in yield, no difference in lodging. So one of the first questions I get in talking to the growers, say, I'm reducing lignin content in alfalfa, and they say, is it going to stand up? Is it going to lodge? So what we find is, depending on which gene you knock out, some of them cause more lodging and some of them don't. This one does not. This is lignin. This is ADL is lignin content, and this is in lower stem samples. This is a 28% reduction in lignin content in lower stems. It's huge. A 24% increase in fiber digestibility in lower stems. So I've been an alfalfa breeder for 30 years, and for most of those 30 years, we've been running a conventional breeding program, breeding for improved fiber digestibility through conventional breeding. In 25 years of conventional breeding research, we've changed fiber digestibility by about 2 or 3%, 20 years. One gene we've got an increase of 24%. I mean, it's a phenomenal, phenomenal advance in terms of fiber digestibility in alfalfa. So proof of concept first is, so we've got this thing, looks promising. You do agronomic performance, forage yield testing, look at lodging, other agronomic traits. Chemical composition through wet lab analysis. You do it on field collected samples over multiple years, multiple locations. Does the trait look stable over years in environments, for example, and in fact reduced lignin does. I talked about lignin content, lignin composition. When we reduce lignin, we see no impact on fiber content, but we do see a huge impact on fiber digestibility. So Dave Mertens at the Dairy Forage Center has been the animal nutritionist that has led that part of the project, and I wouldn't say that Dave's necessarily a cynic, but you know, his first question always has been, "This looks great. When can we feed it to dairy cows?" You know, so until you've got an animal proof of concept, I think there's always going to be some skepticism, especially from animal scientists, which I think is well founded. So we're working with transgenic plants and doing testing. It's all regulated through USDA, and there's a regulated process. We're usually producing plots that at the largest, maybe an experiment that's as big as this room. So when you're feeding dairy cows, all of a sudden you need to have large acres of plots to produce a lot of hay to feed cows. So we put out 6-acre plots of various treatments in 2007 near Boise, Idaho, and harvested enough hay to do a dairy feeding study at the Dairy Forage Center in Madison. I'm sorry, a dairy at the large animal metabolism unit at Land O'Lakes Purina Feed, and a lamb feeding study at the Dairy Forage Center. That was all in 2007. And then I've got a couple of slides of that. Well, I should say, so we had these 6-acre blocks. We went out and when we harvested, we took the samples of the green material. We did, and we looked at fiber content. Again, no real difference between, this is 2 different genes and it's null. So here's the comparison, transgenic versus non-transgenic. No difference in fiber content. You can see the difference in fiber lignin content, and then in fiber digestibility. So what we expected, that's what we've been seeing in field trials over the last several years. So you see a significant difference in fiber digestibility, which is what we expected. So this is at harvest. So this picture was taken the day before harvest. Here's RL1. This is the one that doesn't have any effect on lodging. You can see yield. This is the transgenic versus the control. No difference in yield. Really no difference in lodging. The only difference is in fiber digestibility. RL2 is a little different story. Still no difference in yield, but you can see the lodging impact of knocking out that enzyme in the pathway.
Excuse me.
Yeah.
Is the one in the back a lake or is it ocean?
It's the sky.
Sky.
Yeah. So the blue back here is the sky, and the person in the field there is not terribly tall, but she's probably 5.
We can see the lip.
Yeah. Right. Yeah. So what you can see, I think the important part, you can see how this one's lying down more than the null. So again, the logistics of this is still all regulated. Harvesting the hay, put it in sealed trucks, sending it out to the locations where it's going to be. This is at the Land O'Lakes Purina farm near St. Louis, where they're unloading one of the semi trucks and getting it ready for feeding. This is their large animal metabolism unit. It's one of the few in the country, actually, where they can measure, essentially in dairy cows, everything that goes into the cow and everything that goes out. So it's a very sophisticated animal feeding unit. So you can't see it here, but in back there's a grate. They collect all the feces. All the animals have a catheter. They collect all the urine. Feed intake is obviously measured. Milk production is measured, and so forth. So that was at Land O'Lakes Purina with dairy cows. And I think Dave Mertens at the Dairy Forage Center had, I think, in retrospect, a phenomenal idea. And he said, let's do the same experiment with the same feed stuff and do it on lambs. So the argument is lambs have about the same metabolic needs as a high-producing dairy cow, young lamb. But instead of needing tons to feed a dairy study, you need maybe a couple bales to feed a small lamb study. So he built these units. There's no lambs in the units now, but these were the units that he used for feeding lambs. And we generated a whole bunch of data on both dairy cows and lambs. And I think here's an example. So here's, again, this is fiber digestibility, again, NDFD. Here's the transgenic and the null for two different enzymes. Here's the hay samples in vitro, so that's right out of the field. So that's in vitro, and then these numbers are in vivo. So that's in the animal, what happened when you fed this hay. So what's the disappearance of fiber in the animal. So this is a subset of the experiments that were run. We only had one cow study. They were fed ad lib, and it was part of a TMR. So alfalfa, I think, was 50% of the diet. And then the lambs, we had various studies because we didn't need as much hay. We did hay only and TMR. We did both ad lib and a restricted diet. But the good, yes?
What are the RL1 numbers much for?
It's, well, so the comparison is to its control, right? So these are in different fields.
RL2 is 55, and 53, 56, and 2. They're all the same. RL1 is 53, and then a bunch of 40.
Right. So if you compare this number with this number, you know, so you get the relationship between these two is pretty constant across there. So actually what we find is that if you compare RL1 and RL2 in terms of in vivo, actually RL2 looks a little bit better than RL1. So the good news from this is what we were excited about is that we have a slug of in vitro data, which is relatively simple to generate, and now we've got in vivo data that corroborates the in vitro data. The other thing that this suggests is that lambs are a good surrogate. So, you know, this data suggests that we could do lamb studies and run with the same amount of, with a fraction of the hay, run multiple studies, you know, different feeding regimes and so forth, and get similar results that we'd expect to get in a full-fledged dairy study. So that's not only how much hay is required, but it's also the cost of running the experiments. So one of the things that the animal scientists told us when we were designing the experiment is, if you're going to go through the expense of feeding these animals, make sure you have a big enough difference between the transgenic and the control. So what we did was, starting in early bud, we started sampling every 5 days, and we sampled this is RL1, and here's the null control. And we sampled every 5 days, and what they told us was, when you get a difference in NDFD that's at least 5%, you should be able to pick up an animal response. So we monitored, this is when we harvested. But the interesting thing here is the slope of these lines, which was not expected. So what this suggests is, with reduced lignin alfalfa, quality drops more slowly with advancing maturity than it does with normal alfalfa. So it makes sense, right? You're changing the rate of lignin accumulation in the lower stem, which is what's driving this change in quality with advancing maturity. So that was kind of the first light bulb about this being able to delay harvest. And this is a study at the University of California, Davis. This is in vitro, where they looked at early and late cut. And what they found was, again, comparing RL1 and RL2. With RL2, they had 30% higher digestion. So it's not shown here. This is percent of the null. So if it's 100% of the null, it means no difference between the transgenic and the control. So you can see RL1 is a little bit better than the control. RL2 is a little bit better at early cut. But at late cut, you can see the difference widens. So the later you harvest, the bigger the difference between. So again, reinforcing this changes in the drop in quality with advancing maturity. So in 2008, we produced enough seed to establish a series of kind of preliminary cutting management trials. This is with Dan Understander at University of Wisconsin, Madison, or the plant was at Arlington. But you can see the various cutting management. Some of these were cut earlier, cut later, and so forth. And so the goal was, can we put numbers behind this, different cutting management systems? And in fact, what they saw, this is one of several locations, what we found was you could cut 7 to 10 days later with reduced lignin and get the same quality. So if the control was cut at late bud, you could cut 7 or 8 days later with reduced lignin and have the same quality as the control did at late bud.
Excuse me.
Yes.
Did you get more weight or more production with those 7 to 10 days?
Well, we did, but that's a single cut. So it's probably one less cut per year. So this was based. So actually, we have that data on one cut, and now we've designed experiments to look at a 3-cut regime over a longer rotation. So you could look at the effect over the entire growing season and then multiple growing seasons. So I think the yield thing isn't quite as clear right now as the quality. But
just to understand what the benefit would be on the 7 more days.
well, so too. So we think that there could be a yield benefit. If you cut 3 times rather than 4, for example, there should be a persistence benefit. You know, whether it's in Wisconsin or California, hay producers that are producing for dairy are cutting more often than is healthy for the plant to get high quality. So if you can delay harvest, it's healthier for the plant. You should get higher yield, better persistence. 1 fewer cut is less transportation costs on it, you know, the cost of harvesting. So the other thing, so we actually tried this on for size with dairy farm or with alfalfa producers and asked them, so where do you see a benefit? And what most hay producers said, it's in flexibility and timing. So in the Midwest, for example, hay growers say, I know when to harvest alfalfa, but I'm managing around other farming operations, weather incidents. It's difficult to get out there and harvest on a timely basis. So if the harvest window is this large rather than this large, just the flexibility and harvest timing. Yeah.
Are you going to look at protein later?
We are, right. So
You have to slide.
that's exactly right. So the delayed harvest will result in lower protein content. That's correct.
So in that 15-day window, then, starting at 22,
Yeah, so we actually did it in this study, and I can't answer that right off the top of my head. The, you know, what the dairy nutritionists tell us that oftentimes alfalfa harvested, when you're trying to maximize fiber digestibility, is too high in protein. You know, it's not effectively utilized, it ends up literally getting pissed away. So, you know, dropping from 22 to 19 and a half or something may not be a bad thing, even from a protein utilization standpoint. But that's a great question, and I don't have that at the top of my head. Well, so let me back. So I talked about RL1, RL2. There's 3 that are candidates, and there are different stages in terms of their testing. So these are, I talked about differences of about 20%, more or less, in fiber digestibility, lower stems here. RL3 is another enzyme in the pathway showing over 100% increase in fiber digestibility. Absolutely unreal. It does have so it's getting the right level of expression because it does cause lodging when you express the gene, when you knock the gene down too much. So trying to find that sweet spot's a challenge. But I think that to me, the summary statement is we know now definitively we can use this kind of gene knockout strategy to knock out various enzymes in the pathway, and we can change both lignin content and composition in ways that don't sacrifice agronomic performance, at the same time lead to some pretty exciting changes and advantages in terms of fiber digestibility and the way alfalfa can be managed in terms of cutting management, flexibility in harvest and timing, harvest timing, and number of harvests per season, as I talked about earlier. So the other thing I want to talk about, so that's the fiber piece, and I think I know as a group, we're equally excited about the potential of changing alfalfa protein. Alfalfa produces more protein per acre than any other crop that we can grow in the United States. It's an important source of protein in dairy rations. It's not very efficiently utilized. It gets degraded too rapidly in the rumen, and literally there's a metabolic cost for kind of getting rid of that protein. So I'm going to talk about 2 strategies for managing kind of the protein piece. The first is tannins. So tannins are kind of a ubiquitous phenolic compound found in a lot of different plants. And in fact, there are a number of forage crops that produce tannins in leaves and stems. Birchfoot trefoil, sandfoin, lesbideza. So anyway, a number of different crops. So what tannins do is various types of tannins, but tannins in general bind with protein. And so if you look at the forage crops that produce tannins, what you find is the rate of protein degradation in a rumen is lower in tannin-containing forages than it is in forages that don't produce tannins. I remember early on in the project, one of the scientists at the Dairy Forage Center was saying that he thought if we increased rumen undegradable protein or bypass protein from 20%, which is what's common in alfalfa, up to 30%, it was Glenn Broderick was the one that said this, that we could eliminate the need for protein supplements in alfalfa for high-producing dairy cows. That's $1 billion a year business, feeding protein supplements to high-producing dairy cows and many dairy operations that could be the most expensive feed input in the operation. So to me, managing protein losses or improving the efficiency of protein utilization in alfalfa is the big kahuna. If there's one thing we could do to alfalfa to make it more valuable to dairy producers, it's increasing the amount of rumen undegradable protein. So I don't know if I said this or not. Condensed tannins actually are found in alfalfa. They're found in seed coats and in the tips of glandular hairs, but not in leaves and stems. So again, it's a perfect candidate for genetic engineering. What can we do to change the way tannins are expressed? So the genes are there and the machinery is there. They're just not turned on in the right tissues. So this is a kind of a cartoon that kind of is a sort of an update of some of this tannin research. So if you look at, this is kind of, again, this is the pathway kind of going from the top to bottom. Tannins are made as part of the phenolic pathway. The first thing we need to do is, and in leaves and stems, it's just very tightly regulated. So these genes aren't expressed. So the first thing is increase the flux in the pathway. That's just kind of turning on the spigot so things are kind of flowing through the pathway. These are two transcription factors, which are regulatory genes that essentially do just that. And when you do that, what you'll find is anthocyanins are the first step in this pathway. It's a purple pigment. You express these genes in alfalfa and the plants turn purple. So that says we've turned on the spigot. The next phase is we found a gene that converts anthocyanins into epicatechin, which is one of the monomers. So tannin is a polymer of these two components, predominantly epicatechin, and we found a gene now that converts. And when you express this gene into these purple plants, they turn green again. So it just kind of moved stuff down to the next part of the pathway. And then the last part is condensation of these monomers into the condensed tannins. And I think we're well on our way there, but so that's still the last step in this pathway that's still being worked on. We made a lot of progress in the last this project's only 3 years old, a tremendous amount of progress. I'm confident in the next 6 to 12 months we'll have alfalfa plants that are producing tannins. And this work's being primarily done at the Noble Foundation. Actually, John, I think this is John Grabber at the Dairy Forage Center. I think this was his model. Looking at the whole dairy model for tannin, predicting that tannin alfalfa would reduce protein feed supplementation by 60%, 12% increase in net return for dairy. I mean, what could we do to a forage crop that would have that impact on net return to dairy and increase, and then end losses? I mean, so here, actually both with, decreased lignin means less manure solids. Tannin or PPO, which we're going to talk about next means reducing end losses on the dairy, so significant environmental benefits. And then increasing, obviously, because of the value of either alfalfa silage or alfalfa hay. So a side benefit is tannin-containing forages don't bloat, so there's not a lot of direct grazing in the United States. Virtually all of the alfalfa in Argentina is grazed, for example. Worldwide losses related to bloat are estimated to be in excess of $200 million, so that's a side benefit for tannin. So the other challenge with alfalfa protein is not only is it rapidly degraded into rumen, when alfalfa is used to make haylage, it's rapidly degraded post-harvest. So instead of, in a hay situation, you drop it down to 15 or 20% moisture. Essentially, there's no biological activity. The protein is stable. But in haylage, it stays in an active condition for a much longer state until the pH in the silo drops to a level to inactivate or to stop biological activity. So typically, what we see with alfalfa haylage, up to 50% of the nitrogen is in the form of non-protein nitrogen. So you've already lost 50% of the nitrogen in alfalfa haylage. Oftentimes has already been lost through post-harvest proteolysis. So a group at the Dairy Forage Center found several years ago now that documented that that's what happens in alfalfa. What they found is that doesn't happen in red clover. So what makes red clover different than alfalfa? Red clover has a lot less post-harvest proteolysis. And they found a system. There's an enzyme, polyphenol oxidase, or PPO, that in the presence of a proper substrate forms quinones. Quinones bind with proteins, you get less proteolysis. So that's kind of the general story. And then a group led by Mike Sullivan at the Dairy Forage Center has isolated the gene from red clover, put it in alfalfa, is now engineering alfalfa to produce the right O-diphenyl substrate. So again, this is a project that's come a long way in the last 5 years, looks real promising. So I really see these as really answering two parts of the protein problem. In making haylage, how do we prevent post-harvest proteolysis, or how do we manage that? And then in the rumen, how do we get more of the rumen to bypass the rumen and go directly to the stomach? So there's a lot of people in it. So I'm speaking today on behalf of a lot of different people. The Dairy Forage Center, Neil Martin, who's in the audience, was one of the founders, along with myself and Rick Dixon at Noble Foundation. Some of the Noble people, and there's a lot more than that. The group at FGI, Pioneer, LOL Purina and Monsanto have collaborated or contributed at various stages of the process also. And I want to close with kind of one slide that's just barely outside of this, the realm of what we're doing at the consortium, because everything I talked about is part of this consortium that I discussed earlier, and that's flowering. So what we know is when alfalfa flowers, a lot of be careful I said, a lot of bad things happen, right? So when alfalfa flowers, we start to get a slower rate of alfalfa's growing exponentially. When alfalfa flowers, growth starts to slow down. The other thing that happens when alfalfa flowers, you get this accelerated decrease in quality in alfalfa. So flowering, alfalfa flowers, bad things happen. Yield starts to level off, quality starts to go down. So the nice thing is that flowering in plants is pretty well understood. We know what genes control flowering. We know, I have some idea how we can alter those genes to change flowering time. So I have a friend that's not he's not a plant guy. I'm not a plant scientist, but I was telling him he was asking about my job, and I was talking about some of this. And I said, there's a switch in plants, and it's not just alfalfa, it's for all plants. They're vegetative, and it's all about growth. It's reproductive, it's all about producing seed. That switch goes off, and the plant gets reprogrammed. So he goes, oh, I get that. He goes, I got a 15-year-old boy. And when he was 12, he was growing 3 inches a year. All he thought about was food. He turned 15, all he thinks about is girls. And he stopped growing. And so it's not just plants. I mean, you get into the reproductive mode, and it's a major reprogramming. I think our idea is to use various techniques to delay flowering in alfalfa and continue this kind of exponential growth farther on as a way to increase yield. I think it's the most promising thing we can do to increase forage yield in alfalfa. And then this is in Arabidopsis, which is kind of a lab plant. But here's one of the genes for delayed flowering. And what we find is that when you delay flowering in plants, you get two things. They continue to grow vegetatively, so you get more vegetative growth. That it also tends to decrease the rate of senescence of older leaves, which would be a plus for alfalfa.